Welcome, everyone. We're looking a little different here as we were having some technical difficulties. So you see a small, two of us on the very top and a LambdaVision screensaver. So first of all, welcome everyone to our science track series of the CRF. That took me a couple times to repeat that, to get that one down. My name is Beth Foss, and I am a member of the Choroideremia Research Foundation. I am an affected carrier. I have a father and two sons that have choroideremia. So we just wanted to welcome you here. It's been a little while. We have some more things coming on board, but we just want to say welcome. Hey, and especially welcome to our first-time guest. Jordan Greco is here. He's the Chief Scientific Officer of LambdaVision. It is a company that is producing many things in space and on Earth about artificial retinas. So we're going to learn a little bit more about what that looks like, what their work is doing, and how potentially it could work with choroideremia. Jordan, how are you?
I'm great. I'm great. Thank you, Beth, for the invitation to speak today and the time, everybody's time today. I appreciate the attention and looking forward to talking a bit about our technology.
Us too. Hey guys, I wanted to remind you to please, if you have questions, use the Q&A or the chat feature. That's part of my job. I'll be monitoring that. And as Jordan periodically stops, he'll be just checking in for these questions. So we'll take them as they come and again at the end. So with that, we'll go ahead and get started. Thanks, Jordan.
Excellent. Oh, thanks, Beth. Again, thank you for inviting me to speak here today. I met a few, and I see a few familiar names, I think, in the attendee list. I met a few of you at Arvo this past year. And it's been a pleasure to get to know some of you. And again, I'm just happy to be here to talk more about what we're trying to do at LambdaVision. So as you suggested, Beth, I'm the Chief Scientific Officer of LambdaVision, been with the company since inception in 2009. LambdaVision is developing a protein-based artificial retina to treat patients that are blinded by retinal degenerative diseases, including choroideremia, but our first lead indication would be for patients with advanced retinitis pigmentosa. As I suggested, we emerged out of the University of Connecticut, and this was through the research group of our founder, Dr. Robert Burge, who was a physical chemist, and I'll say a bit more about him in the coming slides. But LambdaVision, just to give you some context, is in the preclinical stage of development. So we're early in development and have not performed any clinical trials to date, but we hope we're leading to that over the next few years. And as Beth alluded to as well, we have our core technology is the artificial retina, but we do a lot of work in microgravity, as sort of indicated on this slide too, with the rocket ship, with the rocket launch on the right-hand side of the image. The talk today is tailored to artificial retina, but and the development and the preclinical development of that technology. But I'm happy to speak a little bit to the microgravity work if it's at all of interest to anyone at the end of the presentation. And with that, I'll get started. And before launching into some of the background on the science, I always like to highlight the career of our founder, Dr. Robert Burge. For some context, he was, he had a varied career and is now retired, but made his career in developing novel bioelectronic devices. And that includes ultimately what we're doing here with LambdaVision and the artificial retina. And I can speak a little bit to some of the other technologies that were developed. They all center around the protein that we currently work with, which is called Bacteriorhodopsin. And he is an expert in the field of light-activated proteins and bioelectronics. LambdaVision, as I said, was founded out of UConn. We're in the preclinical stage of development. We did have a pre-IND meeting with the FDA early on to give us some information and support on our preclinical path, and that's currently what's defining our research efforts to date and over the next couple of years. Our technology is an artificial retina that's aimed at treating retinal degenerative diseases, as I mentioned. we're starting with advanced retinitis pigmentosa as a target indication. But as you'll see, we expect that our technology is a universal treatment option that could be applied to many inherited retinal disorders, dry age-related macular degeneration as another example. And as I think the audience is well aware, there are no cures for these retinal degenerative diseases, and there's an unmet need to restore meaningful vision to patients that are blinded by these diseases. I do see a couple comments coming through. I'm not sure if I'd be happy to answer any questions
You know,
if there
somebody just said, yes, please have him speak about the microgravity work at some
are.
point.
Okay. Okay. All right. I'll be happy to do that. Um, uh, So the artificial retina is a multi-layered thin film that's coated onto a scaffold, and this is an implantable drug product that's inserted into the subretinal space. So the core technology is this protein-coated film that creates a proton gradient upon the absorption of light, and that proton gradient is directed towards the remaining bipolar and ganglion cells of the retina, and essentially serves as an artificial photoreceptor cell layer. There is a surgery that's required to place this film into the back of the eye, and I'll show this in a little more context in a bit. But really, the way to think about us, and I know we're sort of branded as sort of this new approach in the title of this talk. I think we're sort of at the intersection of a couple different um, approaches for visual restoration that you may be aware of. That includes the optogenetics approaches and also the electrode-based prosthetics. We're an implantable like the electrode-based technologies like the Argus 2 and the Pixian Vision or Science Corporation technologies. But we're different in that we use a protein instead of photodiodes or electrodes to stimulate the retina. So, and in that case, we're similar to the optogenetics technologies where they use similar light activated proteins. So we're sort of at the intersection of these two approaches, and have come up with a new way to have this implantable device or drug product that can stimulate the retina. So again, this slide illustrates the core mechanism of our technology. So this is a representation of the retina, a simplified representation of the retina. And as you know, the retina is the light-sensitive tissue that lines the inner surface of the back of the eye. And is comprised of a series of specialized neurons, including the photoreceptor cells of the rods and the cones, bipolar cells, ganglion cells, and that network of neuronal cells is what leads to visual perception. And as you know, the photoreceptor cells are responsible for absorbing light and then transmitting that signal through the rest of the retina, through the optic nerve into the brain. And throughout the progression of inherited retinal disorders, these photoreceptor cells are lost over time. Um, and our artificial retina is placed in place of those cells. Again, it serves as an artificial photoreceptor cell layer. And upon the absorption of light, the light activated proteins in our implant direct a proton gradient towards the bipolar and ganglion cells. And those ions interact with and stimulate those bipolar cells, and that's what leads to the visual stimulation and we hope functional vision. Um, the architecture of our artificial retina is really quite simple in a way. So there are three main components. There's a scaffolding that's really just a thin film. That's a mesh thin film that's ion permeable. And then we deposit alternating layers of a polymer that is positively charged, and and layers of protein, which are negatively charged on both sides. And it's this electrostatic deposition that allows us to create these large scale multi-layered thin films. These repeating bilayers are deposited hundreds of times to create what we have as our architecture for the artificial retina. And then on top of that is another sealing scaffold where we would protect those protein layers for long-term implantation. And that's important to note, and perhaps I should make that clear at this point. This is intended to be a chronic implant, which would ultimately last several years. And you know, and what we'd be interested in is having that be running the course of the patient's lifetime as well. And the protein is quite stable, and I'll talk more about that in a second. Oh, and this is the polymer that we currently use. It's these are active areas of research that we're exploring now. What is the best polymer for our mechanism of action? What is the best biocompatible scaffold that we can use? These are items that we are sort of interchanging and evaluating in our preclinical development. The protein, however, remains the same. That is the active ingredient to our artificial retina, and that's going to remain the same, even though we may deposit those layers in different ways. To say a few words about the protein that we work with, again, the protein is called Bacteriorhodopsin. It's very similar in terms of its structure and function to rhodopsin, which is the visual pigment found in the photoreceptor cells, but there are some key differences. So Bacteriorhodopsin, like rhodopsin, is a 7-transmembrane alpha helical protein. It is found in an archaeal, excuse me, an archaeal organism, which is a single-celled extremophilic organism that's typically found in salt marshes. And this protein is expressed in the outer cell membrane to facilitate the production of chemical energy for the organism. When this protein is expressed in the cell membrane, it forms a very stable semicrystalline structure. So the proteins are arranged in trimers. So there's 3 subunits of proteins that are compacted together, and then those trimers are arranged in this lattice type structure. And this is often referred to as the purple membrane. And this has been studied. This is a material that's been studied since the 1970s when it was discovered. It's like its name suggests, it's a vibrant purple color. And the integration of the protein into this lattice is really what leads us to be able to use the protein in non-native environments like the artificial retina or other technologies that have been developed by Dr. Burge, the founder of the technology. So there's a long history of that kind of work that I can speak to if there's any questions about it. And the protein also has a very large dipole moment, meaning that when you apply a voltage, the protein orients reproducibly in one direction. And this is important for us as we are making thin films based on this protein. Again, the protein is well characterized in that it can facilitate proton pumping, which is its inherent activity in the native organism, and also has been shown to facilitate electron transport for other types of devices that are harnessing that energy from the photochemistry of the protein. On this slide, this is really quite complex, and this again has been studied for many years. This slide focuses on really the photochemistry of the protein, what happens when it absorbs light, and why it's useful in these bioelectronic devices or the drug product like the artificial retina. Really, the take home on this slide is that when the protein absorbs light, it undergoes this proton pumping mechanism. And again, the proton pumping is what we rely on to interact with the remaining bipolar and ganglion cells. So this protein cycles where it resets after 20 milliseconds or so. So it can absorb light, pump a proton within 20 milliseconds, and then it's able to absorb light again and undergo that process again. So this cycling effect is what we utilize in our artificial retina, and can continuously pump protons in response to incoming light, incident light. And again, there's different photointermediates that are represented throughout that pumping process. There's a light activated chromophore that's really responsible for absorbing light. It's called all-trans retinal, which is vitamin A. That's linked to the protein, and that's what drives that activity. And the protein absorbs quite broadly in the visible range of the visible spectrum as well, which is another feature that suggests it's useful as a a biomaterial for the artificial retina technology. And as I mentioned, the protein is highly stable, can resist temperatures upwards of 90 degrees Celsius, and it can undergo this cycle millions of times before degrading, again, leading to a benefit to use in the artificial retina technology. Are there any questions at this point?
There is. We had a person come in and ask, at what stage is this implant beneficial? Is it beneficial with partial vision loss or only at full vision loss? And I might tack on an age that you're thinking about.
Yeah, no, that's a great question. Right now we're targeting advanced degeneration. So I guess in that sense, it would be for individuals that are they're almost completely blind with bare light perception. Um, the idea being that we wouldn't want to damage any remaining
Mhm.
vision that may be there through the surgical procedure that's required to insert the artificial retina. Um, I think that's something that's, an area that we'd want to refine and perhaps, would want to help individuals sooner than that, of course, before it's an end stage type of um, solution. But currently where we are and how we're modeling our preclinical strategy, that would be the patient population that we'd be targeting first.
Okay. Thanks, Jordan.
Thank you. Um, and like I said, I could talk for a long time about this, but this slide highlights some of the other technologies that were developed based on this protein throughout the career of our founder. I just find it interesting, and I think there is sort of a direct path to the development of these other technologies to the artificial retina, which we began to develop in the early 2000s. But again, this protein is so unique, and it's been applied in optical computing technologies. Like I said, the protein can act as a semiconductor and can be useful for energy storage applications based on its photovoltaic activity. So there's really a lot of robust research around this protein that we utilize as we tailor its use in the artificial retina and figure out how to harness that activity most appropriately for stimulating visual function. Um, just briefly, I mentioned this earlier, and again wanted to give some context. And this is just a broad overview of our development timeline as for context for the upcoming slides. But we are again in the preclinical phase of development, and I'll describe a bit about where we are now and what we've accomplished to date. But currently, we're really focused on improving the manufacturing processes of our artificial retina. That includes making the protein, but also it includes the layer by layer mechanism that we use to create the multi-layer thin films. And then we're really focused on testing these films in animal models. For efficacy, we're utilizing rats, and for safety studies, we will utilize pigs, and that all is of benefit to lead towards our phase 1 clinical trial, which we expect to begin on the tail end of 2027 and into 2028. The protein, again, is something that we harvest from the native organism, Halobacterium salinarum. It's an archaeal organism that's found in salt marshes. And this is something that we work with in our own labs. We use these large incubator shakers shown on the left-hand side there, where we grow this organism and ultimately harvest, the purple membrane fragments from the cells, which contain that Bacteriorhodopsin. And like I said, the purple membrane is an appropriate name for this material. That you can see the vibrant purple color of the protein in solution in the center image there. And this is something that we're continuing to optimize and develop. And we're currently working with a contract development and manufacturing organization, CDMO, out in California, called List Labs, to help us develop this process in a way that can be performed with good manufacturing practices or GMP, where we can produce this material for a clinical product. So we're that's where a lot of our effort is right now. We've been making some great progress there this year that I'm excited about, and it's something that will continue as we need steady state production of the protein for our preclinical and clinical efforts. The artificial retina itself is made using a layer by layer electrostatic deposition process. So this is done in a number of ways in our lab, which are described on this slide. But ultimately, like I showed in the preceding slides and with the architecture of the artificial retina, we the structure is such that it's alternating layers of a polycation and the protein. And again, we need hundreds of those layers in order to generate the signals that are required for visual stimulation. So we achieve this over time using a dip coating method, as shown in the center image there. We've pivoted towards using a fluidic chamber type of device where we have a scaffold sitting in this reaction chamber, and then the various solutions are passed over that film to create the multi-layered structure. And that's currently where our efforts are, especially as we consider, again, doing this in a way that's relevant to GMP practices for clinical products. The layer by layer method is quite tunable, and we can control it in a number of different ways. This is where a lot of our the early years of our preclinical development were centered. We can change the number of layers, and that directly leads to different optical densities or different light absorbing properties of the artificial retinas. And that also seems to directly be related to the activity of the artificial retina. How strong is the signal that we're creating, which I'll show on the next slide. We're also able to choose materials that are biocompatible. The image on the right-hand side demonstrates that we have been able to show viability of artificial retinas that are several years old, in this case up to 6 years old, where the absorption spectrum of the protein contained within the thin film has remained ultimately unchanged, which is a good sign for its stability for use over a long period of time. And we always are trying to improve the stability and performance of these films, but so far we see a lot of promise in the way in which we're creating these films, whether it's dip coating or through the fluidic chamber approach. And I could speak. I'll kind of throw this out there now, and we're exploring this layer by layer assembly method, as alluded to, in a microgravity environment as well to strengthen that process even further. But I'll say more about that in a bit.
Hey, Jordan, we had a question come in.
Yes.
You baited us well on that one. Here's the question, and maybe you want to address it later on as it's in your slides. But looking at the process itself, what do you expect to be the biggest hurdle to get to clinical trials? And then the writer said, other than the FDA, ha ha ha.
The biggest, yeah, I mean, we're sort of to be perfectly honest, there are many challenges that I guess we all face that we're developing products that are intended for clinical use. The manufacturing consistency and analytics surrounding all that has been a challenge for us, and continues to be something we have to continue to develop and come up with new methods. As you maybe are understanding, the artificial retina is quite unique in some ways, and the protein also is quite unique for to be used on an implantable material. So we've had to develop a lot of our own analytical tools to understand quality for both the protein and now the artificial retina. And that's been a big area of our internal development. As a chemist, it's been a fun area of the development for me. I enjoy that part of it. But there are limited off-the-shelf analytical tools that we can use to assess the quality. So that's a big part of our internal research now. And then certainly, the non-clinical work that's forthcoming for us. Uh, the 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 safety studies in pigs, for example, there's a lot that leads up to that in terms of the bioanalytical assays and planning and making sure that those are executed properly.
Certainly.
So yeah, I don't know if I answered that perfectly or specifically enough. I guess my answer is everything's challenging, but we're with the right people, and that's the other thing. We are a relatively small group here, but we've over the years accumulated a wonderful team of advisors and consultants and
Good.
that are helping guide us through these challenging processes. So
Certainly. We do have another question that came in. It's
sure.
again, if you want to incorporate it later into your talk, Jordan, you can. The question though is how well will this restore vision? Is it restoring it completely or partially? And then will it be able to restore both night vision, day vision, replacing both cones and rod functioning?
Mhm. Yeah, no, those are all great questions. I think, ultimately we think that we have a lot of benefits compared to some of let's say, the electro-based technologies or uh, some of the gene therapies that have been developed, and in the sense that um, the architecture of our thin films can be generated so that they are intended to be high resolution. The scaffolds that we select, there's certain a density of apertures or holes in that scaffold to make them ion permeable. That's what drives the resolution. We think that with layering the protein in the way that we do, we think that we have an, the advantage of utilizing the size and scale of the protein to make really high resolution films. But what will a person see is difficult for us to understand at this point we have theoretical models of that but I hesitate to take that full leap showing you this is exactly what someone would see. Um, but so far, what we've determined, and I'll show a little bit more of our preclinical data
Mhm.
in the next few slides. We're seeing that we have control over that spatial sensitivity and that the temporal sensitivity of the artificial retina suggests that we can have functional vision. This will be targeted for the foveal region of the retina, underneath the foveal region of the retina. So in terms of night vision and color vision, and we're not again, we're not so sure what exactly a person would see outside of what's been observed in some of the electro-based technologies. Um, and I think generally speaking, those have been, high functioning and high contrast environments. And I think there has been some development on being able to assess different colors um, and operate under varying light intensity conditions. And that's something we've tested here too. And I could show some figures on that.
We do have another question that came in, but I'm going to let you continue, and we'll ask this question the next go around.
Okay, just feel free to interject with it at any time.
Okay, you bet.
Um, uh, right. So again, we use layer by layer to create our films. And to one of my comments to the questions, we've been developing in-house techniques to assess what those films look like. And this is one example of that, actually, where we have developed uh, a method using confocal microscopy, which is essentially a method that uses fluorescence microscopy to look at three-dimensional structures or structures in 3 dimensions. So we can use this method to look at the thickness of our films and the homogeneity of the protein deposited onto those films due to the autofluorescence of Bacteriorhodopsin. And this helps us understand, okay, what is the percent area coated? Is this region of a film better than a different region of the film? How uniform is that thickness? How is the protein coating those apertures that are, and you can get a sense of the um, material. So this is a mesh film that we use as a scaffold. In this example, you can see those fibers, and we want to coat over those fibers, which is shown with increasing numbers of layers here uh, from the uh, left-hand purple image, which is 25 layers, all the way up to 200 layers. You can see how those layers accumulate over those pores. Um, so this is again a method that we recently developed, and we continue to enhance those imaging technologies to tell us how what the quality of the films are. And this also relates to the activity of the films too. This is one example of how we're measuring activity. We're using the photovoltaic activity of the protein, as I mentioned, where we can um, directly measure the proton pumping across the film of the multi-layers. So a proton, I misspoke there a little bit. It's the proton pumping occurs per protein, but we're generating an ion gradient or a proton gradient across the film based on the many protein molecules that are integrated into the architecture. So we can monitor that proton gradient, and we've seen with some of the photocurrent measurements, as shown in this slide here, there's an increasing signal as we increase the number of layers that are deposited onto the film. And this matches with our expectation on that very well, and is something we continue to explore. I mentioned that we have done some ex vivo extracellular recording experiments as a proof of concept of the capability to stimulate degenerated retinal tissue using our artificial retina technology. This is where we're working with the rats were P23H rats, which are a model form of retinitis pigmentosa. The rats in this case, their retinas were excised and placed on top of our artificial retina thin films, and we tested the capability upon the absorption of light to stimulate those cells, and we measured ganglion cells, the activity of ganglion cells, as we shone light on that assembly. So we were able to see that with increasing light intensity for 200 layer films, we were getting very high activation efficiency, meaning that we would see a signal for every pulse of light as we increased in that intensity. And to a lesser extent, with 100 layer films, that we got about half of that activity, as you might see in the left-hand image there. And then when we did control experiments where we either flipped the film or had a blank scaffold, in this case, these are films are flipped in the opposite orientation. We see little to no activity that occurs when we're directing light towards the artificial retina. So this was some of the early proof of concept work, and this speaks a bit to the light sensitivity question that came up about night vision versus daylight vision. We think the light intensities shown here are relative to indoor ambient light conditions on the lower end of the x-axis. This is something that we're still evaluating. How sensitive are these protein layers, and in what context? And will light amplification be necessary once placed in an in vivo environment? Those are all sort of open questions that we're evaluating, but it is an area of active research for us. In addition to the extracellular recording experiments, we did additional testing with a multi-electrode, where we had an array of electrodes where we can test the spatial sensitivity of the films. And with this study, we were able to show that we can selectively activate areas of around 200 micrometers in diameter, which is about the feature size of some of those electrode-based technologies, or at least some of the early ones. And like I said, we think that our resolution is far higher than we've been able to determine so far based on the feature size of the scaffold and the protein. But this is something that we continue to test with the various analytical methods that are under development with LambdaVision. We've also done some in vivo work where we have inserted the artificial retina thin films into rats. this is leading towards some efficacy testing where we hope to look at varying doses of or numbers of layers, um, and look at the capability of restoring vision in these rats using electrophysiological techniques like ERG and visual evoked potentials. And this is something that we're launching into a bit more now, um, late this year and into early next year. But we were able to do some pilot surgical methodology studies where we were able to show how we can get this into the back of the eye. This is an OCT image of showing that the film, the arrow is pointing to a film that's inserted into the subretinal space, which is the intended position of the artificial retina. And then we also looked at this against some controls where we had a sham procedure where we did not insert any implants, where we had an uncoated implant. Um, these materials were all tolerated for a short term, up to 6 weeks. And we hope to extend that to much longer testing in the coming months. But so far with our current evaluations, we've haven't seen any major source of inflammation and with any of the materials that we've implanted. And we've also done some of the surgical development in the larger animal model, which will be the model for our non-clinical studies, safety studies. This is in pigs. So we similarly did a 6-week study. And the intent of this study was more how would we get this into the back of the eye of humans? And the surgical procedure was developed. This helped us hone what the size and shape of the artificial retina could be. That's something that is quite adaptable. We can make this any size or shape that is necessary, and maybe at some point we would have patient specific uh, 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 morphologies with the artificial retina that's punched out of larger sheets. This is something we're still working on, but it is the preliminary information tells us this can get successfully we successfully inserted this into the subretinal space of these large animal models with some promise for our upcoming studies. And that's circling back to some of the our development timeline here, which is again is very, very broad, and there's a lot more detail that I can get into on what we're doing and when we're doing it. But for the sake of this presentation, I just want to get across that we're probably within the next 2 years, we'll continue our manufacturing efforts and some of these efficacy and safety studies. We do have some funding to support up until 2027 to support all this work, which we're excited about. And I think it positions us well to continue our path towards getting to phase 1 clinical trials, which is, of course, ultimately the goal.
Hey, Jordan, I think this is a really good point. In time, I can ask this question that was posed. Endpoints, right? That's something that we have as a foundation. We've kind of focused on in previous studies that we've been involved in. Which endpoints would be used or defined as key efficacy measures for the trials?
Mhm.
I'm sure those are still being developed, but you want to talk just a second on that?
Yeah, at least sure. No, thanks for the question. I think we to date what we've been able to utilize in both the rat and the pig studies has been um, mainly electrophysiological endpoints. So we utilized electroretinography, ERG. We also tried working with visual evoked potentials as well, VEP. Um, and have done a little work with optokinetic measurements as well. So the sort of in concert with we hope to use these methods in concert to understand what the visual function is of these animals. I think we're also exploring the use of multifocal ERG to look at spatial sensitivity of the films as well, and again isolate the activity within the retina. So it is an active area of our development as well, honing these methods as it specifically relates to our films, which certainly is a challenge in some ways. We're working directly with a group in North Carolina called Powered Research to do some of the to do the rat studies that I mentioned to test efficacy. And these, where we have been with them the past year or two is really looking at those methods and the endpoints and understanding how can we use this to make sure that this study is giving us the data that we need and helping us understand the activity and helping us understand what, if any, modifications we should make to the technology. So that's a core area of our focus now, that you've struck upon there, and
Mhm.
it is, an active area, as I said.
Cool. Thanks.
Thank you. Um, yeah, and actually, I was just wrapping out this portion of the talk. I just wanted to acknowledge our team. Nicole and myself have been with the company since inception, as I mentioned, and have a long-standing relationship with studying Bacteriorhodopsin and these devices, in addition to our founder, Dr. Robert Burge, and it's um, something we remain very enthusiastic about, and something that we're 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 here for the long haul in terms of getting we've made so much progress to date, and there's still much to do, of course, in the preclinical development, but we see a lot of promise in what we're doing, and we really you know are excited to the coming years and getting, making more progress with the non-clinical work, and again, really reaching towards that phase 1 clinical trial, and we work hard every day to try to make that a reality. And we have a great team of board of directors here, with a lot of biotech experience, and Connecticut Innovations, and Susan and Kirk have been wonderful advisors and have quite varied careers. Both have varied careers in the biotech area. Wanted to also highlight our research team, Dan, Krishna, and Ashley, who are the ones in the lab every day, performing a lot of the research, internal research and development. And we've had a lot of great support from many advisors. I just highlight a few here that have been critical for our development to date, but there's many more I could highlight. Dr. Del Piore, Chief of Ophthalmology at Yale, is on our scientific advisory board and has been a wonderful supporter. Dr. Modi has assisted us with a lot of our regulatory communications and our strategy moving forward. And Scott Rudge has been critical in our manufacturing efforts as well. And Jack is a wonderful business advisor. Happy to pause again here to take any more questions. And as we alluded to before, I can say a bit about our microgravity research as well once those questions have been answered, if there's still time.
Do you
But
know what? There aren't any questions right now. So maybe you could just go ahead and it was very thorough, I think. Well, now we say that and one comes in. Here we go. Have you started to use AI, LLM, artificial intelligence, yes, large language model in the research? Do you think that AI, LLM might accelerate the research and testing?
yeah, I think I generally, I think we utilize it in a simple manner in terms of our, internal R&D. It's not something that has been a critical component of our day to day, I would say, in terms of, the critical path of, the study designs and things of that nature. Certainly not opposed to it, and it is something that we're looking at, I think, more and more. And I think we're having my attitude about that is, if that's the direction the field is taking, I'm certainly amenable to that, and I will work closely with our advisors and to help implement anything that might be a good idea for us to implement. But I have to say, I'm not a heavy user of AI in general, in both my personal and professional life to date. And maybe that should change a bit, and I should pivot to that a bit more, but nothing specific to really that's noteworthy at the moment, other than, we have eh, we have done some work with AI and machine learning related to our manufacturing methods. I guess that's the one exception to date where where we're trying to use imaging, and you can I mentioned that we create the films that we create are hundreds of layers. 200 layers is our current target. Um, that takes a long time to make. Typically, it's a several day process. And so any way that we can automate that and use AI to help assist and make adjustments to improve the thin film quality, um, and how we can utilize the imagery that we capture over that manufacturing period. That's an area that we've been developing to help us automate and improve quality. So I expect that we'll continue with that a bit as those methods mature. Uh, so
We
uh,
have about 10 minutes. And for those of you that can't see it, there is a NASA on the bottom of your screen. So I think you should. And again,
okay,
I'll pause you if we have more questions come in, folks. So if you do want to ask more questions, and you might get more after this.
sure. Yeah, feel free to interject. And I put this at the end because I wanted to make the first part of the talk concise and focused. But certainly, if you visit our website and you see a lot of what's going on with our research and publications and such, we do have quite a bit of support from NASA as indicated on our funding slide that I just showed as well. The idea behind this work is that the layer by layer processes that we've developed over time, we think could be improved in a microgravity environment, such that in a microgravity environment, we reduce some of the negative effects of gravity that could impact film quality. And this has been observed in a number of different fields where they look at making protein crystals in solution, or they look at different three-dimensional assemblies in microgravity to have just more ordered structures, more stable structures. So without just relying on diffusion within microgravity allows us to make, in theory, a more homogeneous film, one that isn't affected by convective turbulence within solutions. And we expect that these films will be of higher quality. So NASA, we have NASA's support in exploring this over time, and we've been looking at this for a number of years now, and have done 9 missions to the space station to look at the layer by layer process in a microgravity environment. This is, I'm sure the question you have in your minds is, does this mean you have to manufacture the artificial retinas in space in order to advance your technology? And I think the answer to that is no, not necessarily. I think a lot of what we've learned through this research and development is applied to what we do on Earth. We have created layer by layer devices that function both on Earth and microgravity. Um, and it's sort of a parallel path that we're exploring in different ways. And ultimately, we just want the highest quality film possible. If we were to find that the microgravity environment is necessary for making really high quality films, well, that's something that we can explore, and the feasibility is something we're also figuring out as well alongside NASA. How can one make a product in space, and that aligns with their initiatives as well. So we've this slide highlights some of the I guess some of the key learnings over time and what we've been able to achieve. There's a manufacturing device that we've developed alongside our implementation partner, Space Tango, where we can't use a dip coater, as you might imagine, and have open beakers to dip the films into. We need to use this fluidic chamber style approach where we have a scaffold and then solutions are passed over that film, and then the contents of those solutions are deposited electrostatically onto the film over time in the same way that we do in our labs here in Connecticut. So we've been able to develop this hardware that's been quite reproducible to date with a high degree of automation, and again, building in this is where some of the AI and machine learning comes into play, making this automated, using imagery to help detect faults or you know different events that might occur during the manufacturing process that need to be redirected. And this is all done in a way that could serve for GMP for clinical products. Again, we're not there yet, and neither is the ISS facility, but it is something that again the feasibility is being tested. And this slide shows some of the films that were created. You might be asking yourself, what is this strange shape? Uh, what is this strange geometry? This is a reaction chamber where we deposit the thin films, and this is what would serve as the artificial retina, although we would punch out um, individual implants along the long axis of this chamber. And that's why that's shaped in that way and it really just comes down to what facilitates the best fluidic motion throughout these chambers. So we've been able to consistently do this and manufacture the films in this way, and it's something that we continue to explore. Again, I would say in a parallel path to a lot of our preclinical development, even though a lot of our key learnings are directly a benefit to what we're doing in our labs in Connecticut every day for the development of our artificial retina. And something that I think NASA is very interested in is, while this has relevance to our artificial retina technology. As I mentioned before, there's a number of other applications that we've explored with Bacteriorhodopsin as a photoactive material. But also, there's a number of biomedical technologies and other advanced materials technologies that use layer by layer. So a lot of what we're learning from the hardware standpoint and the analytics could be directly related to other technologies outside of what we do at LambdaVision, and that's something we're trying to leverage just from an R&D standpoint and sharing our know-how to others that might benefit from this methodology. So I think that's all I had regarding the microgravity work, but certainly happy to answer any questions on that as well. It's certainly a fascinating area of research, and it is exciting to be in this field and being able to influence, some of the future, what NASA is looking for and, of some of the commercial space stations that are being developed. Yeah, and again, if this directly benefits the artificial retina, that's something that we're, of course, excited about too.
That's just a little George Jetson kind of work, right?
So, it's sometimes I have to pinch myself and really just take a step back because it's a lot of, it's just fun to be a part of, honestly. And then a lot of the work that goes into the launches and the missions on the ISS are just, incredible to see the teams come together and make it happen, make it a reality. Um, so it's great and uh, has helped us immensely as a company to date. So
We have some more questions.
sure.
Can you answer them for us?
Yeah, absolutely.
Good.
I'll try.
So next, what is your target for the life of the transplant based on a person's age?
Yeah, that's a great question. I think that's something we're still testing from a stability standpoint, but what we know about the protein and its lifetime and its photochemistry, we expect that the artificial retina could last up to 10 years. Now that doesn't directly relate to a person's age, I would say, so um, uh, I I think that would be true of any individual that would receive that film, whether they were 20 or 80. But I think, we're still testing that lifetime. There's a possibility that we can, there could be multiple implantation events that could occur. That's something that's down, you know, years from now for us to evaluate, but it is something we are considering. So that's currently where we stand. And, the in vivo testing needs to prove that out, of course. We've done only short-term tests in an in vivo environment. In vivo is very different than in vitro, which is where most of our testing has been. Of course, we have to mitigate any sort of immune response that might occur, which is something that we know and we're working towards.
Here's another question. It says, hi. So if I understand correctly, There will have to be a decision between your protein-based artificial retina and the electronic neuron implants, right? Isn't it possible to combine them both? Thanks in advance.
Yeah, no, that's a great point. Some of the early, well, there's some great work that's been done with those electrode-based technologies. The Argus 2 certainly paved the way for a lot of folks that are even not developing the similar technologies. They've done just their path in terms of uh, uh, what they've had to experience on the regulatory side of things. And what they've developed technologically has been incredible from my perspective. And I know that there are still groups that are developing electro-based technologies like Science Corporation and which was the Pixium group. Um, some of the early concepts of our artificial retina actually were what you're suggesting is sort of a hybrid electronic, bioelectronic um, architecture where we would harness the photoelectric activity of the protein and create these pins that would interact with the retinal tissue. Sorry. Um, so it's not off the table, and I think there's a lot of key learnings from the electro-based technologies that we still rely on and are inspired by. And the architecture of our films have changed over time and could find their way back to that architecture potentially. But right now, the ion mediated approach where we have you know, a mesh scaffold is our current approach. And we see advantage to not having any external components. That's an important point, I suppose, as well. Our film is a standalone film where it would be inserted into the subretinal space, and that's it. There's at least currently, no external components, glasses, and any penetrating wires or anything like that would be necessary.
Mhm. As a follow-up to that question, he writes, thanks for your reply. Do you also plan to implement your research finished products in Europe at any point in the future?
Yeah, certainly. I think our intention would be to do our phase 1 clinical trial in the US, but we do I think it would like to pivot as soon as possible to the European patient populations as well. There's no direct strategy for that, I would say, at the moment, other than we would plan on doing that as soon as possible.
So Jordan, when you get to your clinical trials with your advanced RP patients, are you seeing it basically staying in the Northeast, or will you have several sites as you're developing it further? Where do
I
you
would
see?
expect, I'm sorry.
No, no, yeah, go ahead.
I would expect it to be over several sites in the US. But that, I don't think we would even try to relegate it to just the Northeast. So I think obviously we still have a lot of planning to do there, but I
Right.
don't see it limiting us in any way there outside of it likely being in the continental US, I would imagine.
That's all the questions we have. We're gonna wait another minute for one last straggler to come in. They usually do. But thank you for your time. Thank you for
Oh,
all the work that your team is doing. Even though, this is something that we hear a lot. You and I spoke about it when we met last week or talked last week in preparation for today. I think so much of what the foundation's responsibility, and it's in our mission statement, is to provide education as well as encouragement, in terms of what's coming. And boy, you're certainly doing this. This is really exciting. Lots of hope for the future for sure.
well, thank you. No, it was fun talking to the group, and there was a lot of great questions. I appreciate the opportunity, and it's something, like I said, I mean, we're just very enthusiastic about this technology. I, I'm have a personal connection to the research, being so involved in it for a number of years now, and we're a very close-knit group here too. We just are just have our heads down and are trying our best to bring this forward. So like I keep saying, there's plenty ahead for us to work through, but we feel like we have some good positive momentum now, And I hope that we can stay in touch with you and and your team and the folks on this
Absolutely.
call and keep you updated on where we're headed.
Well, we're going to count on that
All right.
for sure. All right. Well, again, we always want to make a plug for this too, folks. If you are not involved in the CRF, the Choroideremia Research Foundation, please become a member. This is like the best way to get involved, learn about the science. We try to keep it at a layman's pace. And as you learn more, you can dive in deeper. We have a robust YouTube channel, and on our website, you can watch many of these science videos. But please become a member and continue our work with your donations for science. So thanks, Jordan. Have a great rest of your week.
Thank you. You too. Thanks, everybody.
Thanks a lot, everyone. We'll see you next time. Bye-bye.